Thanks very much for sticking around. Like I said, it's a very long day. Look, in the interest of proper disclosure, since it's popped up a few times, I should point out that a long time ago, around about 8 years ago, I did receive funding from Gatorade. I was part of a team that investigated hydration status in cricket bowling accuracy, hence why Australia won the Ashes. I should say to counter that, though, I have recently received non-industry funding to study the benefits of removing sugar, so that should counteract the Gatorade contamination. The other thing I should point out too is 7 years ago, I was drinking 2 litres of Coca-Cola a day on average and weighed 92 kilos, and I now don't drink any and weigh 75. Thank you. Thank you. I should point out, Coca-Cola wasn't the only thing I cut out. I changed a number of things. I lecture in exercise physiology and biochemistry, and my key research interest is in understanding how cells regulate whether or not they store or burn / utilise fuels. I tell you that for 2 reasons. If Rick thinks he just gave you biochemistry, you haven't seen anything yet. I will do my best to walk through it as simply as possibly as I can. The other reason I tell you that is to point out I'm not a cancer researcher, even though I'm about to give you a 15-minute talk on sugar and cancer. I point that out because I am probably about to make something which is obviously quite complex come across relatively straightforward and simple. However, if some of the current discussions on cancer are true, I might not need to be a cancer researcher to be qualified enough to give you this talk. And I'll give you a reason for that. There's been a lot of interest in the last couple of years at least online, if not in some published case studies, on the effect or potential link between sugar intake and cancer incidence. Epidemiological observational studies showing cancer incidence increasing with obesity and type 2 diabetes. A recent study showing that type 2 diabetics on metformin, which reduces blood glucose, tend to have lower incidence of cancer than diabetics who don't have metformin and therefore have higher blood glucoses. If you do a Google search for cancer and ketosis, you'll find a number of TED/TEDx talks about cancer survivors talking about how they've beaten it with ketogenic diets. You'll also come across a number of esteemed professors who are doing proper research into the ketogenic effects or the beneficial effects of ketosis on cancer. I think one of the things that really jumped out to me during my readings over the last year as I've got more interested in this was this particular quote I came across on a blog. I admit it's not a peer-reviewed piece of evidence, but I found it astounding when Rob Wolf came out with this one statistic that exposes what an abysmal failure war on cancer has been, is that the death rates from cancer today are just as much as they were 50 years ago. And when you consider the billions of dollars and man-hours have gone into, or man and women hours have gone into researching cancer, it does suggest that we've missed something. And that's what I want to focus this 12.5/15 minutes on here. And that is, what is it that the cancer community potentially could have missed? And I guess to address that, we need to think about what is it that they've been focusing on. And when you read most of the literature that comes out, there's an overwhelming feeling and obvious thing that cancer researchers for at least the past 50 years have considered cancer as being a disease of genetics, and what some people refer to as the somatic mutation theory of cancer. And that's the one where you think of an environmental toxin that gets into your cell, damages your DNA, and therefore that destroys some particular function. Now, most cells can regulate their growth and also their death in a tightly regulated process, and one of the mutations that commonly people say toxins will do is it stops cells from being able to destroy themselves when they proliferate uncontrollably. And so then what that leads to is further oxidative damage within the cell, which damages more DNA, and then you get these cancerous cells with multiple mutations in them. And the Human Genome Project was supposed to tell us what the key to the genetics of cancer was, and therefore help us identify cures. Unfortunately, what it appears is that around about only 12% of cancers are actually genetically inherited. Also, when people go screening mutations or DNA of cancer patients, reportedly the mutations are very random. And so therefore, except for a handful of mutations, there's not that much consistency between them. However, the important thing to note is this somatic mutation theory of cancer isn't the way it always was. In actual fact, 90 years ago, there was a German biochemist by the name of Otto Warburg who viewed cancer as a metabolic disease. And around about 100 years ago then, the environmental toxin that caused cancer was sugar. Now, Otto Warburg won a Nobel Prize, not for this cancer work, so he wasn't any slouch. And depending upon which biographer, historian, or Wikipedia reference you go to, he was also nominated for at least 3 other Nobel Prizes. Now, what this does do is it drives a paradigm shift that's occurring at the moment within cancer metabolism, or cancer research, I should say. If we think about cancer as a metabolic disease, then we start looking at completely different targets for therapeutic agents. And I should point out that if you really want some good, hard reviews, not hard reviews, easy to read reviews of the hard science, you do very well to go searching for any of these particular professionals. Thomas Seyfried from Boston College, Dominic D'Agostino from University of South Florida, I think it is, amongst the others, who are publishing and have research teams working and looking into this area. What I wanted to do was at least give a bit of a brief background as to the metabolism processes that they're referring to, so that perhaps then if you do go do those readings, you've got a little bit of a platform to come from. Okay, so when we talk about cancer as a metabolic disease and we start thinking about the role sugar might play in it, everybody thinks for the first purpose of energy. And so if we think about what the role of glucose as an energy source for a cancer cell is, we need to have a little bit of a walk through what does it mean for a cell to get energy. Now, you've heard this thing called ATP bandied about a bit today. You've also heard mitochondria bandied a bit about. I'm going to give a little bit of an image to maybe walk us through it. Here's a representative cell. It could be a generic, it could be any cell. I've simplified it down as much, so we're just going to assume that it's just our basic structure of a cell. The black ring is our cell membrane. Cell membranes are made up of fat and phosphate. And embedded within that plasma membrane are a couple of key functional proteins, such as your cell receptors, which will respond to hormone-like, or there might be some transporters, nutrient transporters. They allow things to come in and out. Very simple. Now, when we talk about the energetics of a cell, it's not about carbohydrates, fats, and proteins. From a biochemistry point of view, it's all about ATP. If you want to contract your muscle, it's not a glucose molecule sitting at your actin myofibril filament, it's an ATP molecule that you release the energy from to power that work. If you want to transport something across the membrane, it's an ATP molecule that gets hydrolyzed to generate that energy. So when we think about cell energetics, we want to think about ATP and where are we getting that from and what's it particularly used for. It's then when we have the next layer, we start talking about individual macromolecules. So if we've got glucose, it comes in through a specific glucose transporter, and it will go through glycolysis. Glycolysis, in its most simplest form, is we take glucose, which is a 6-carbon ring, which Rob showed you some pictures of, and we rip it in half into 2 3-carbon rings, and that's what we call pyruvate. 2 molecules of pyruvate, every mole of glucose that's going through. Now, when we make pyruvate through glycolysis, we also make a little bit of ATP, which is the energy source for the cell, and we also make this thing called NADH. Now, NADH is what we refer to as a reducing agent. Really what we're worried about is the NAD bit. That's the molecule itself, and it holds on to electrons. And in biochemistry, electrons or oxidation reactions are hydrogen atoms. That's what the H is representing at the end there. Now, when we've made a lot of pyruvate, we've got really 3 options as mammals. For a yeast cell, we'd ferment it into ethanol, and we would then be able to harvest that to drink and what have you. As mammals, around about 1% of all the pyruvate you make gets converted into alanine. The rest of the pyruvate has 2 options. It enters this part of the cell called the mitochondria, where it gets converted into acetyl-CoA and enters this thing called the Krebs cycle, also referred to as the TCA cycle, also referred to as the citric acid cycle, all depending upon the country and origin of the textbook that you're reading through. All the TCA cycle does is takes our acetyl-CoA and amongst other things producing carbon dioxide produces lots more of this NADH molecule. The NADH molecule reacts with this component called the electron transport chain just to regenerate NAD, and that's in a closed loop. So as long as you keep putting acetyl-CoA into the system, you'll make more NADH that will regenerate NAD through the electron transport chain. Now, if we add oxygen to the mix, we make ATP. This is what we call oxidative phosphorylation. So this is the 2 different processes by which we can get ATP from glucose. Substrate level or cytosolic glycolysis, where we make pyruvate, we get a little bit of ATP. Or if we're in the presence of oxygen, we go into the mitochondria, we make lots more. Something to identify is if we're just using glucose in the cell, we're getting very small amounts of ATP, 2. If we put it through the mitochondria, we can get lots more, 30 moles of ATP. And so if we're thinking about an efficiency level thing, you'd be like, right, if I need lots of ATP, so let's think that I'm a cell that's uncontrollably replicating and I need lots of energy to fuel that, I should be using the process by which I get the most energy, which would be mitochondrial oxidative phosphorylation. Now, in exercise science, there's always this dogma, oh, when you run out of oxygen or you don't have enough oxygen, you produce lactate, and lactate makes your muscles hurt and that makes you fatigue. Problem is that's not necessarily true. It's not when you run out of oxygen. Your muscles can be saturated with oxygen. It's that your mitochondria can't use it as much, or they can't use it well. Or another way to think about it is you're producing pyruvate far quicker than you can convert it into acetyl-CoA and put it through the TCA cycle. Now, under those circumstances, something happens. Pyruvate doesn't enter the mitochondria anymore. It stays in the cytosol and produces this thing called lactate. Now, lactate doesn't give us any more ATP. But what it does do is it rips off the hydrogen off your NADH, regenerates NAD, so that we can continue going through this glycolytic pathway. That's how we get our energy from glucose if we do or don't have oxygen available. A fairly complex, but I've tried to simplify it there. What about fats? Fats are relatively simple. Fats enter through a fat-specific transporter. There's no oxidation or metabolism of fat within the cytosol. They enter into the mitochondria, get converted into acetyl-CoA, and then we see more ATP. Fat's slightly different to glucose because it's got more carbons. We can generate far more ATP. So if we go through oxidative phosphorylation, we're now into the hundreds. What about protein? Well, if we're having protein or amino acids, they enter through various transporters. They also go straight into the mitochondria. And whether or not they create ATP is up to debate depending upon the type of amino acid, the condition the cell is in. But most people wouldn't be too upset if I just said that their major role is making sure there is a TCA cycle for acetyl-CoA to enter into. So it's called anaplerosis, and we mostly use that. We'll get into different amino acids. That there is cell energetics in 5 minutes. If this is being recorded, I'll let my students this semester have get access to it, and it'll save me 3 weeks of lectures. Okay, so what does this mean for Warburg? What does this mean for cancer? What does this mean for glucose? Right, so Warburg, looking at cancer cells. Now, Warburg won his Nobel Prize for identifying regulatory roles of the electron transport chain bit. So while he was doing that work, he also noticed one other major thing. If I have cancer cells growing in a tube and leave them be with various fuels, they generate heaps and heaps and heaps of lactate, thousands of moles of it. And so people were like, right, well, that obviously means to us then that they are using anaerobic glycolysis or non-oxidative pathways to generate their energy. So let's test this. Let's give them lots of oxygen and let's see if they start using fat or they stop producing lactate. It doesn't work. So a cancer cell can be sitting in an environment that's saturated with oxygen and they still generate buckets of lactate. It's a phrase termed aerobic glycolysis in most textbooks, or what is more appropriately coined nowadays as the Warburg effect. And it's quite a reproducible effect across most cancer cells that they show excessive rates of glycolysis in the presence of oxygen. So this is what a cancer cell looked like to Otto Warburg. Mitochondrial dysfunction, meaning that they are only using glucose as a source when oxygen was present. So Warburg made the Warburg hypothesis, not to get mixed up with the Warburg effect. Warburg effect is glucose being converted to lactate in the presence of oxygen. The Warburg hypothesis was, well, then the prime cause of cancer is the replacement of the respiration of oxygen with the fermentation of sugar. He said his cell looks like this. Cancer is caused by some respects where we have dysfunctional mitochondria, so therefore cancer cells can't use fat, they can't use proteins, they can't use ketone bodies as a fuel source, they can only rely on glucose. So this is 90 odd years or so ago. Now this is the thinking that has become, experienced a little bit of a renaissance at the moment. And a number of people have been looking for this, and sure enough, when they've looked at various cancer cells, they're noticing quite a few pieces of evidence to build up on this hypothesis. And that is that even though glycolysis is a relatively inefficient supply of ATP, it becomes a great source of ATP because you can do it really, really fast. And you can do it much faster and without a dependency on oxygen transport. And if you think about masses of tumours, tumour masses of cells, which ultimately start working in some hypoxic environments, you don't need the oxygen to generate ATP if you're using a non-oxidative pathway. And some people have recorded the rate of glycolysis up to 200 times that which you would find in a normal cell compared to a cancer cell. Mina Bissell from Berkeley, I think it is, has shown that in breast cancer cells, they've got 400 times the amount of glucose transporters than in non-malignant breast cancer cells. And so she bases a lot of her conclusions around the fact that if we take a cancer cell here, if it's got 400 times the amount of transporters on it compared to a non-malignant tumour, what we see is a cancer cell being flushed with glucose, which would drive the glycolytic process, providing energy. Now, when you have a cell undergoing extreme amounts of metabolic activity, the other thing you produce is a lot of free radical or oxidative stress. And that would add to further mitochondrial damage. And so what we get is a few pieces of the puzzle falling into place. There is a slight problem, though. 2009, that Vanderheiden paper, is the one that most people refer to as being something that put the nail in the coffin on the Warburg hypothesis. Not necessarily the Warburg effect, but Warburg hypothesis. It's interesting because it's mostly a review article and it actually refers to other papers that have shown that many cancer cells still have actually perfectly functioning mitochondria. There's nothing wrong with them, or there might only be minimal things wrong with them. So the mitochondria can still work within a cancer cell. There is still some mitochondrial respiration, but the rate at which we're getting ATP from glycolysis is still much greater than the mitochondria. What other people have shown as well is that when we think about the Warburg effect and Warburg hypothesis, there are a couple of other things. People who have starved cancer cells of glucose do show the cancer cells die, but they don't die because they run out of ATP. They die because of oxidative damage and oxidative stress to the tissue. The other thing people have shown is that we can feed cancer cells fat and they will grow. Not necessarily saturated fat though. Omega-6 polyunsaturated fatty acids will stimulate a cancer cell to grow and can be used as a fuel source in certain cancer types and certain papers. But this is not necessarily a problem. This is only a problem for the people who believe, I'll pick up on a debate from earlier, that a calorie is a calorie. Right? For people who think all energy is the same, this is a problem. But as a biochemist, a calorie is not a calorie. Energy is not the be-all and end-all. It's what you use that energy for. It's where it comes from. If it was all about energy, then perhaps there is a problem. But a growing, proliferating cancer cell has needs that extend beyond just an ATP source. What we do know is the aerobic glycolysis, or the Warburg effect, is a reproducible finding in cancer cells. But if we rely only on ATP as being the be-all and end-all of cell replication, then it falls short. A cancer cell has a requirement to make new cells. To make new cells, you need to make new plasma membranes, which means you need lots of fat, lots of phosphate, and the proteins that sit in them. The core of a cancer cell that's replicating is DNA. So you need lots and lots of DNA to be made. And I notice conveniently there's a picture of DNA over here, luckily, because I don't have one in my slide. Here's the DNA, and if we just think about it, these are nucleotide bases, and this is a sugar backbone, a ribose sugar group. And the cancer cell has to make millions of these cells so it can continue its proliferating effect. And that's where we probably see sugar having a much more greater role on cancer metabolism than just as its energy source. We've come back to this picture. We know that we've got glycolysis occurring at greater rates. We're producing hundreds of ATP, hundreds of pyruvate, hundreds of NADH now through glycolysis. We've still got our mitochondria. We know that they are working, and there is some oxidative phosphorylation taking place. But there is an addendum onto this that we need to put. Not all the acetyl-CoA goes through the TCA cycle. In the cancer cell, the acetyl-CoA gets pumped back out of the mitochondria and sits in the cytosol. We know that roughly 90% is the number that people say, 90% of the pyruvate that's produced actually ends up as lactate. So there's still around about 10% of the pyruvate that is tracking into the mitochondria. What we do notice as well, though, is that there's greater rates of the pentose phosphate pathway. You might also see this as known as the pentose phosphate shunt, but since I get excited when I talk and have a tendency to trip over my words, I like to avoid any chance of being inappropriate. The pentose phosphate pathway is the major role by which we make this thing called ribose 5-phosphate. Ribose 5-phosphate is the sugar backbone of DNA and RNA. The other thing we make from the pentose phosphate pathway is this thing called NADPH. And NADPH is a highly effective reducing agent, which would protect cells against high oxidative damage. The other thing that NADPH is essential for, stimulating the synthesis of fat from acetyl-CoA. So now what we have occurring here within our cancer cell is by having high rates of glycolysis and excessive uptake of glucose, we're not just using it as an energy source, we're using it as the fundamental building block for making more cells. We can't use fat in the pentose phosphate pathway. We can't use ketone bodies in the pentose phosphate pathway. We don't necessarily can't use protein within that. We need sugar for that. So for a proliferating cell, you need sugar to supply two of the major arms that you need. Fat to go into your plasma membrane, DNA so that you can continually replicate. There's one other key fuel for cancer cells, glutamine. And many cancer cells will utilise glucose or glutamine as their predominant fuel sources. Now, a lot of glutamine enters into the mitochondria to prop up the TCA cycle to make sure that can cope with acetyl-CoA that's coming through and supply energy. But around about 60% of the glutamine that comes in also gets converted into lactate. If you convert glutamine into lactate, you also make NADPH as a byproduct. And NADPH then can act as a buffer against oxidative damage for the cell that's got this high energy production, and the NADPH can also then go and stimulate your fat synthesis. That's an abridged version of cancer metabolism using sugar. Now, again, I point out it's simplified because I haven't even talked about the influence of hormones. But don't worry, I'm not going to, not today. For those knowing who was going to be in the audience today, I thought I'd better point out where does fructose fit into the picture as well. And if we look at pancreatic cancerous cells in particular, they tend to grow better on fructose than they do on glucose. And people looking at pancreatic cancer cells have shown that the fructose, it can enter glycolysis at its usual midway point, but it also acts as an excellent substrate for the pentose phosphate pathway and can stimulate then further ribose production for DNA and for fat synthesis, NADPH for fat synthesis. Okay. I know I've rushed through that, but I noticed we were 40 minutes over time, so I thought you wouldn't mind if I went quickly and finished it early. So I guess in conclusion, for anybody who's going to find themselves in conversations about the growing attention on the role of sugar in cancer, the first thing you can do is you can at least tell them you know who Otto Warburg was now. But also point out that it's not necessarily all about using sugar just as an energy supply of ATP. But in actual fact, if we take certain cancer cells and give them sugar, they use it almost exclusively to provide the platform for making more cells, the core of cancerous tissue. You can use it as ATP in glycolysis. It will help in the production of DNA. It will help in the production of NADPH for the fat that they need for their plasma membrane. So, of course, what does this mean practically then? Does this mean that if we just go on a carbohydrate-restricted diet, do we cure cancer? Do we offset it? And the answer is Sometimes yes, sometimes no, we don't really know. If we have a look at small animal models of the mouse and the rat, then we get mixed results depending upon the type of cancer and dosage or the type of dietary intervention. What's happening at the moment is more and more human case studies on ketogenic diets that are coming out being used as a combined therapy with chemotherapy, radiation therapy. And that is that while we're using chemicals also at one point to attack one part of the cancer, using a dietary treatment that should be effectively starving them of the nutrients that they crave should be working out as well. And that seems to be where people are moving in the future. And that is looking at combined therapies of dietary treatment along with cancer, along with drugs. Some people, I should point out, if you go and Google those individuals I had up before, are actually working on that aspect of having a look at designing drugs that attack those specific individual parts of a pathway. You could alternatively just remove the start, which would be the sugar. And I just thought, I won't go through these individual, but I can leave these up here now if there's any questions so you can get photos or write them down. But if you're looking for peer-reviewed, published papers on the role in which carbohydrate restriction or ketogenic diets may assist in the treatment of cancer patients, these are probably the top 4 or 5 that you should go and have a look at. Thank you.